SINE, a representative from SINE and from Haystack Analytics. So would you like to start first, Anirvan? From SINE, we have Mr. Rakesh Rajiv, who is the Senior Incubation Manager. SINE is IIT Bombay's business incubator, stands for Society for Innovation and Entrepreneurship. And another short talk will be delivered by Dr. Anirvan Chatterjee, who is the co-founder of Haystack Analytics, spin-off incubated at SINE.
Check. A very good afternoon to one and all. I'm so pleased to stand in front of you because I remember the last time I stood here was 2 years back when we were celebrating the 15th year anniversary of SINE. There was an event called as Dialogue with Stakeholders, DWS, which we did. And that was 2 years back. And now SINE is Society for Innovation and Entrepreneurship. We are the technology business incubator at IIT Bombay. After I've been told that there is a slight delay in the slides to come up. So yeah, 15 years, 2 years back with COVID, etc. We are 18 years now. We were established in the year 2004. And most of the work that we do in campus is to identify good startups, not just from campus, but also throughout India. We have different models by which we work with them. And I'm joined here by my CEO, Poini Bhatt, and my colleagues, Bratati Mukherjee and Pranita. So over the last 18 years, we've been incubating startups day in and day out. That's our job, making sure that we get access to good quality startups, train them, connect them with different industry partners, tap into the resources which we get from campus and outside. We connect them with different people which can help them in their journey, and we become part of them as well in their journey. So in terms of the support that we provide end-to-end support to our startups right from the stage that they're at a very early stage. When they come in with an idea, we provide them with various government grants, thanks to the government taking the first risk, because our clients are startups, right? And statistics say that 9 out of 10 startups fail. And government taking that big risk at that stage has helped us as incubation centers across the country. And SINE has been blessed to, I can say proudly, that 80% of our startups are successful in terms of the statistics, which is over the last 18 years. We have incubated 197 startups, of which 150 are still operational and doing well. And so in terms of the support, so end-to-end support, which I mentioned. These are the different support that we provide, ranging from infrastructure, mentoring support, that is what startups value the most. And being in IIT Bombay, it's like a gold mine. We have access to talent right from faculty members, students, the various connects which we get, and events like these, wherein we partner with different entities within campus. The incubation support is for a period of 3 years, wherein we provide different kind of support. And the next slide talks about our impact. I mentioned 197 startups. And during COVID, we were slightly worried with the number of folded companies. Those are startups who didn't sort of take off. Will that number increase? But it was heartening to see that just 1 or 2 numbers got added to them. And not just that, they didn't completely shut down. Some of them repurposed themselves to support for various COVID-related activities. Again, the government pitched in there, and there were different grants which the government gave for supporting such startups. One ratio which I would like to point out to is on that second square there, the ratio of SINE to external funding, 1:180. So that basically means that for every rupee, every rupee that SINE invested in, our startups have went on and raised 180 rupees. So I mean, I often tell startups are our heroes, right? And I have a few of my heroes sitting here in the audience as well. There's Anirvan from Haystack, and there is Sai Prasad. Sai Prasad was here a few minutes back. Okay, so we have a couple of our startups over here as well. And you know, we had the opportunity of having the first unicorn from an academic incubation center when one of our startups, Gupshup, became a unicorn when they raised 100 million from Tiger Global. Now coming on to the healthcare space, if I dissect the entire 197 startups, 48, you know, roughly 25%, 1/4 of our startups are in the healthcare domain. And that really goes to show, again, from various funding sources, especially the BIRAC, Dr. Satya was here in the morning, how BIRAC has sort of contributed. And even in campus, we have different centers such as WRCB, Betik, Tata Center, all supporting affordable healthcare sort of startups. So if I take the impact of healthcare-related startups, as I mentioned, 48 of them, currently we are incubating 32. So when I say incubate, there is also another program wherein we provide them with support, like government funding coming through. All put together, we have 32, and of which 19 are revenue generating. For healthcare startups, especially, it takes time for them to bring to the market. But when I say healthcare, it is across different domains. I can share a few examples in the future slides. And again, funding raised by startups. In India, especially healthcare, most of them are in the hardware sort of domain. It's not easy to get funding, but our startups have been able to raise that kind of funding from different sources. And this is the next slide is the team which really makes it possible. So we've been expanding quite rapidly. So when I joined SINE 7 years back in 2015, we were a 9-member team. And now we have multiplied 3x, which also goes to say that we're also shifting to a facility which is 3x our current facility. We have a center of excellence building coming up on campus, and the inauguration will be done in this very room next week by R.M. Chatterjee. And some of our facilities, I'll just quickly run through. We have a bio lab, metalworking lab, a fabrication lab, etc. Some of our partners that we work with across corporates, government, international, etc. And with corporate, we engage on 3 different models. One is in terms of the program engagement. We run corporate accelerator program with our corporate partners. Validation partners, we have Sanofi, which is a pharma company, helping our startups with mentorship, Catapult from Mahindra, etc. And CSR engagement from our CSR donors. Now, as I mentioned, these are our heroes, really, right? So we have MedPrime Technologies, which are students from Professor Debjani Paul's lab. They came back and started off in SINE, the company called as MedPrime. They make this mobile-based microscope, which is really portable, and we use them in our biolabs as well. We have Indimension, which works on medical image, AI on medical images, which helps radiologists to identify if there is something wrong with if there's a condition which can be missed by the naked eye. It's an assistive device for helping that. We have ImmunoAct. So this is a relatively new company, less than 3 years old, started off by a faculty member called Rahul Purwar in Bioscience Department on campus. Indigenous CAR-T cell therapy for cancer treatment. So CAR-T is well known, but the expenses are prohibitively high for the developing countries to afford. He has made that possible, and clinical trials are going on at TMH, Tata Memorial Hospital. With that clinical trials, 3 patients have been declared cancer-free using this technology. Haystack, we have Anirvan who will be speaking more about this. We also have Sai Prasad from Pacify Medical. I'll just quickly welcome him onto the stage so that he can quickly show his device. Basically, it's a device which can help in reducing the time for wound healing in chronic wounds.
Thank you, Rakesh. So this is a simple portable surgical gun, which is used by a plastic surgeon to spray skin in real time on wounds. And currently, Indian Air Force has shown interest in this. And we have done some validation studies at Sassoon, where patient has been healed just by spraying skin without the pain of the stitches. So this is, and I'm one of the incubators supported by SINE and supported by different grants from BIRAC and currently by Defense. Yeah, thank you.
Thank you, Sai. And we also have Anirvan. So Haystack Analytics is working on genomics. Genomics, at one point in time, when I interacted with them 6 years back, I had to refer what genomics was. Right now, because of COVID and other reasons, we know what genomics is. So using genomics, they help in diagnosis of infectious diseases such as TB, etc. I'll call Anirvan to speak more about this.
Now, while this is getting set up, thanks Rakesh for the kind introduction. Thanks Devanshi, Abdur, WRCB team, Professor Devjani Paul, Poini, Pranita, SINE team, always they have our backs as we move along. So thanks a lot. I'll probably just wait for this to come up. We should have activated Windows. I think given that the entire thing is about low-cost diagnostics and affordable healthcare, there might be some skepticism as to what is genomics doing there, but clearly genomics is not affordable. And that's where I think I started the whole thought of why should genomics not be affordable? And also the question is to what is the total cost of healthcare? And I think that's something we should keep in mind, because if the diagnosis is done right, then the cost of healthcare is much lesser. The cost of healthcare increases because of the journey of the patient. I think Rishabh was here. His study that we did on journey of a patient was something very interesting. So to start off, I think what I'll do is I'll play this video.
So now, if we see, there's a hint there. This patient was brought to my center with the right pneumothorax. On sputum examination, he was AFP positive. On CBNAT, he was R sensitive. Now, I went according to the government guidelines, and this fits into a first line sensitive category of patients. See that out of the first line drugs, out of the 4 drugs, this guy is sensitive only to rifampicin, and the rest of the 3 drugs he is resistant to. So if you go by the government guidelines, you would have actually missed this case and treated this case as a first line sensitive patient, because we are just doing the AFP there and the rifampicin sensitivity by the CBNAT.
So I played this example for a couple of reasons. One, this is a doctor in Meerut. And the patient is of tuberculosis. Neither the patient can afford genome sequencing, nor this doctor has ever been exposed to genome sequencing. Today, we are really proud to say that we are serving about 10,000 PIN codes in this country, and we are providing genome sequencing-based diagnosis for tuberculosis, which is cheaper than culture DST and microbiology. Genomics can not only be cheaper, genomics can not only be accessible, it is something that is more comprehensive and prevents every such patient to not take antibiotics which don't work on them, and stop the spread of antibiotic resistance, and stops us from replacing a treatable epidemic with an untreatable epidemic.
So now, if we see, there's a hint there.
I'm sorry,
this patient was brought to my center with the right pneumothorax.
So what we have seen in the field, and there are countless such examples that we have, is how doctors have been able to use genomics. And just to put it in perspective, what we do and how we have enabled this, for all the people here who are looking into data and looking of AI and the scale of data, genomics is expected to exceed all the data that we know today in the world. So storing of the data and processing of the data is one of the greatest challenges that we know today in healthcare, because everybody needs genomics. We kind of understand that in clinical medicine, genomics is probably going to play a very important role. But the entire bottleneck is on data analysis and clinical decision. And the ripple effect of that is, for that very reason, genomics is more expensive, genomics is less accessible, and therefore genomics is used lesser and lesser, where genomics should be used more. And this is the Haystack solution. By integrating the entire analytical platform and actually create walkaway automation on genomics, today we are probably one of the rarest company in the world where at an operational level, we have 0 human beings sitting between raw data and a genomic analyzed report. Every report that goes out today is analyzed by a software. And while AI and ML, and there are a lot of things that people talk about, what really matters is, when we went into the field, our theory was, and this is what we think as entrepreneurs. And I think probably in one of these rooms, I was told that it's not that the product will sell because it is great. The product is great because it sells. And when we went into the market, we just wanted to test that hypothesis. If we want to make genomics accessible, then there are companies who are already accessing the doctors, the hospitals, the patients. Will they take up our solution? 9 months down the line, we are probably working with everybody in the country today. The biggest benefit of that to people who have done DNA sequencing will know, now we are able to actually collate a large number of samples in a single sequencing run, which means our turnaround time is shorter than most microbiological and genetic technologies, which also means our cost for each sample is probably the most affordable that it ever has been. And all of them are able to use our technology to produce reports which they brand themselves and take to the market, to the patients and the doctors. For the first 3 months, we supported them in reaching out to the doctors. All our partners are now doing their own marketing and use genomic solutions. We are really proud to say we probably created the largest number of genomic diagnostic providers in the world in the shortest amount of time. What we have been able to also, thank you, what we have been also able to do is we started with the government support, because we're working in tuberculosis. There's no point doing anything in tuberculosis without government support. And that went a long way. What we have been able to now expand this entire thing into is, we have expanded drug susceptibility testing from just tuberculosis to every bacterial infection and also fungal infection. And we have changed the model where we have now taken the onus of bringing sequencing on the hospital's table. Till now, the sequencing was being done by the diagnostic labs. They collect the samples, do the sequencing, and send out the results. We now want to bring sequencing to the hospital's table, especially for cases where even 6-hour delay in sample transport renders the test useless. And this is the test that we are working with our partners. I think so some of our partners are sitting here, and we are really grateful for them, to them for identifying and working with us. This test is now for all bacterial and fungal infections. It is able to detect any bacteria or fungus in blood, along with antimicrobial resistance, in a time span of around 12 hours in the hospital setting, because it is based on the nanopore sequencing platform. So we have actually integrated our kit with the nanopore sequencing platform with the compute device, which means today, as we speak, in several hospitals that we are working with closely on the clinical performance and all of that, there are at least, to my knowledge, at least 30 people who have never done anything other than a COVID PCR, who are now doing genomic diagnostics for sepsis cases, where we are able to identify bacterial and fungal infections in less than 12 hours. That's been our journey. As rightly pointed out by many people, and also certainly here, it is a stellar team that I work with. It is certainly a team where I am not only the I'm not only the least smart, but I'm also the least useful. We have a lot of people from IIT Bombay here who have done PhDs in some of the labs of professors sitting here. We have got, again, the kind of support that we have received. Our earliest backers, SINE. I remember Poini told me very long back, "Kya genomics bol raha hai, samajh mein nahi aata hai." And then she said, stop working with the government, get into the private sector. So all of these advice that we get, we tend to ignore them and then realize they're good advice and then reap the benefit of following them up after 9 months. So their support, we've got support from Dr. Velumani, who was one of our earliest investors at Haystack, and he's continuing in our second round of investment. Also, tremendous support from the Mumbai Municipal Corporation. They're probably, in my opinion, they're probably the most experimental municipal corporation that I have come across. They were doing COVID sequencing before INSACOG started doing COVID sequencing. And we were doing it in a lab about 100 meters from here. The lessons that I have learned is, no matter how difficult it seems, everywhere, if you keep asking, there are a few people who will support you along the way. Many of them are in this room, many of them are not in this room, but I thank all of them. And most importantly, I thank the team who, in a startup, spends more than 18 hours a day working, and I wish I could let them not work for a few hours in the day. That's my current aim right now. I want the team to work slightly less. It's not happening. So thank you.
Hey, thanks Anirvan for the wonderful talk. I just want to know, want to get your thoughts on what is the bottlenecks to bring the genomic test to the home at a cost that everyone can afford.
The question is, why do we need it in the home? I'm just trying to think, because genomics intrinsically is supposed to be comprehensive, which means the biggest strength of genomics is data. It also becomes one of, so actually I'll tell you what, when we were doing tuberculosis, we had the option of taking the test to every general physician. We didn't. We took it only to pulmonologists and chest physicians, because I don't think a general physician has the ability to prescribe bedaquiline and clofazimine. So that's the big question. Do we need it at home? Probably not. Do you need the data on your handset? Absolutely, yes. So the cost of genomics is not expensive at all. I mean, I've heard these things several times. DNA sequencing is not expensive. Getting the sample to the DNA sequencer is difficult. And that's the only job that we need to do. So I think in less than 3 years from now, DNA sequencing is close to being cheaper than TrueNAT right now, if we want it that way. So you definitely can get the DNA sequencing data on your handset in a private and secure manner. You definitely are looking at a sub $100, in Indian rupee terms at least. In 5 years, you'll be looking at a less than 4,000, 3,000 rupee price point for whole exome. Bacterial genomes are going to be even cheaper. So yeah, that's coming. It's not a question of if, it's definitely already here. There are a lot of companies who are working towards bringing it closer and closer to near to point of care. The question that we will probably be pondering with most is whether and where we should have that genome sequencing data accessible, because even today, ACMG says that exome data should not be acted on by a user themselves. I think that's the responsibility that we have. But cost and accessibility is not going to be a problem. Certainly not with companies like Haystack.
Thanks.
Yeah, we have a question.
There's one question here. talking about genomics being cheap, so I have a question. So here doing genomics of bacteria or prokaryotic organisms. So if you take it to cancer genomics, is it possible to become it cheap or is it some way?
Good question. So it's not cheap per se, it's inexpensive. So that's why I put a caveat. It's nothing is, I mean, if you anything that costs more than 15 rupees is difficult to recall as inexpensive. But the cost of healthcare actually comes down if you do the genomics well. Coming to cancer, I think it can be inexpensive again in terms of the total cost that a patient incurs if the diagnosis is wrong. But the technology per se, again, it will depend on how many cancers and how many. So, okay, cancer is quite complicated when it comes to genomics because then there is companion genomics and there is detection genomics and all of that. But if we can again build a largish number of samples into a single sequencing run for a set population, it can be. So the cost of sequencing is today 500 rupees per GB. That's the cost of sequencing per GB of genome data. Thereabout 500, 900. Don't hold me to it, but thereabout. And the amount of data that you require for some of the DNA sequencing is not more than 3 GB. The challenge becomes when you start looking at RNA sequencing, and that is where I think a lot of the challenges that the world is working on today in cancer and other infectious and non-infectious diseases also, because we think RNA sequencing, isoform sequencing, these are going to be some of the mainstays going forward, and they are awfully expensive right now. But scale, once applications are there, costs come down. I'm not always worried about the cost. The value has to be right. If a doctor doesn't put it on a prescription, that test has no value. So the more number of tests that can go onto the sequencing platform that a prescriber can write in will bring down the cost of all the tests. But it is possible. Cancer sequencing is also inexpensive if the samples are there. The difficult part is RNA sequencing, isoform sequencing, and so on and so forth.
All right, thank you. I think there are more questions, but in interest of time, maybe we can take them offline if that's okay. We'll do the poster session now, which will be on the ground floor foyer.